Morning. First of all, thanks very much to the organizers for accepting our work, DIMet, an open source tool for the differential analysis of isotope-labeled targeted metabolomics data, which is the fruit of cooperation between the cancer biology and bioinformatics teams at the University of Bordeaux, France. I will start this presentation with an introduction, followed by the brief description of the technology of labeled metabolomics. And then I will explain the expected characteristics of a tool for performing the differential analysis of this kind of data, a use case, and I will end up with the implementation, conclusions, and how our work can be improved in the future. Our teams work in glioblastoma metabolism. This is a very aggressive tumor despite heavy treatment. And there is a link between the exchange of certain metabolites between the tumoral cells and the other non-tumoral cells present in the microenvironment. And we specifically use omics to identify changes in lactate-related metabolism. We obtain transcriptomics, and on the other side, we obtain targeted tracer metabolomics. We separately analyze both types of omics, and at the end, we want to integrate them to be able to draw conclusions at the system level. Targeted tracer metabolomics, or stable isotope-resolved metabolomics, or labeled metabolomics, all synonyms, as you may know. And this technology helps us to understand the dynamics of the metabolic pathways at the system complexity. To do so, cells are fed with stable labeled substrate. For example, in this figure, I'm showing glucose, which is marked in the central chain by carbon 13. And we follow the propagation of this labeled substrate across the different reactions of the system by measuring the isotopologues. For those that are new in lab techniques, isotopologues are the different species given by the number of marker carbons in the molecule. And as I am depicting here for citrate, we can express these isotopologues in percentages to be able to decrypt the behavior of the reactions. For example, if nothing gets marked, maybe there is a block, or if everything goes to market very quickly, we can also do another different conclusion. So we have from labeled setup to a dual information given by the total abundance metabolite, total abundance that we can obtain by summing up the abundances for the individual isotopologues. And we have the information of the labeling patterns that is proper to this label technique in comparison to the classical approaches. And what happens often in the experimental settings is that we have conditions or time points, and we need to be able to compare and identify changes not only for groups of conditions, but also in the time course setup. That means that we need to perform a differential analysis. Once we have all our data and or dual information, we proceed to look for a tool. And in the ideal case, we need this tool to be open source, capable of doing comparative and time course analysis, which has solid statistics, which is fast, accessible for non-developers, that has also functionalities for the labeling information and not only for the abundances that are conventional. And now I am thinking that there is one other point that is missing here, that is a multi-platform, because most of the mass spectrometry world relies in Windows, but new software is available in many other Linux or Mac OS systems. As I have listed these best practices expectations for the tool that we need for the differential analysis of labeled metabolomics. And has been big efforts into this direction. And I am showing in this slide only a very small subset of the tools that are available to perform such analysis, but I'm not going to go into details for each one of them this time, but just saying that none of them fulfill completely all the criteria that I have listed before. And that's why we have developed DIMet. But instead of showing all at once how our tool works, I will illustrate how can we use its functionalities in a biological use case. As I explained at the beginning of the talk, there is a link between metabolism, glioblastoma impassivity, and in our previous EMBO molecular paper that has been published recently, there has been shown that the knockout of LDH A and B genes is associated with a lack of conversion from lactate to pyruvate, and this step is crucial for cell to adapt to the environment and ensure the invasiveness. The experiment consisted of different knockouts and the control. These cultures were subjected to different oxygen concentrations up to hypoxia at 48 hours. For this presentation, I will only focus in the experiment, in the part of the data comparing the double KO and the control. And in the complementary setup, the malate aspartate shuttle was blocked. This shuttle is a special series of reactions closely related to TCI cycle and amino acids. And this experiment uses cycloserine to block the shuttle in a time series setup. And I will be showing the treated group. First, with our DIMet tool, we were able not only to visualize, but also to statistically analyze the abundances that I am showing here in the first part of the analysis for the double KO versus control samples. And we analyzed all the metabolites, but I am only going to show here the most relevant findings in the context not only of the statistics, but also the network. And alpha-ketoglutarate showed a highly statistically significant increase in its abundance. And even if for glutamate, the increase in abundance was not as strong, we went to the isotope and we could identify that there was a predominance of marked species for both metabolites that was statistically significant. And in the data set, there is also another metric that is the mean enrichment, that is a weighted mean of the isotopologues abundances. And we have in the differential analysis of this labeling part, statistically significant decrease in the labeling. So we can hypothesize that something is happening. Something can be, for example, the accumulation or just a hyperproduction, and to be able to elucidate which of two possible hypotheses are plausible, we also used transcriptomics differential analysis. In our case, we used this tool to identify the genes that were statistically deregulated, and we could be able, with our tool DIMet, to integrate the transcriptomics and the labeled metabolomics results from the differential data into a metabologram. The metabologram shows the metabolites in the left and the transcripts on the right. And in the center, there is the average fold change for both omics. And at the TCA cycle, and also at the amino acids metabolism level, we could see that these perturbations were coherent with a severe speed decrease in the reactions from alpha-ketoglutarate to succinate and glutamine. And we could project this information from our metabolograms to the the pathway directly to be able to have the view of the reactions happening. In the time course analysis, we could extract the top relevant changes in time, and we found that for the glutamate and glutamine, the expected behavior was observed. It was the increase in labeling, also in abundance. And at the level of malate and aspartate, we observed that they had an accumulation of non-marked species for the first one. And for the second one, we were in the case of a decrease in abundance and an increment in labeling that was maybe due to residual activity of the transaminases, but it's still current with a good blockage of the system. That was intended to be demonstrated. So the functionalities globally for OTool are divided in the exploratory analysis by graphical representation of the data, a statistical analysis that are both univariate or multivariate. In the univariate group, we have the ability to compare between two conditions, to perform time course analysis automatically, and to perform multi-group analysis as well. For the multivariate, we have the principal component analysis, and finally the integration via the metabologram that I just explained. Now the question is how to use OTool. How can we profit of its functionalities? Well, we have already our version in GitHub, but even better than that, we have packaged our tool in PyPy catalog, and we just have a very standard installing that is cross-platform, because via virtual environments we can also in Windows do a setup for making it work. And we have also examples that we furnish in Zenodo to understand the configuration and the data folder that needs to be organized in a specific way to perform our analysis. And the advantage is that we can keep trace of our parameters and the data that was used with this way of working. And we are working currently to offer to the community a web interface via Galaxy Europe that is ongoing and has the advantage that the user can just by the graphical interface provide all the parameters without configuring the files himself. Internally, our work or tool has a very complicated, kind of complicated structure, but I am schematizing very simple way here that we use our configuration manager framework given by Hydra, which is a very up-to-date way to work for different parameters. And our processing modules and visualization modules rely in trustful Python libraries. In conclusion, our tool has fulfilled the expectations, which is open source, capable of doing comparative analysis, has solid statistics, is fast, accessible for non-developers, and the functionalities that we include also consider the particularity of labeling information. DIMet is the first one-stop tool dedicated to the downstream analysis of labeled Isotope-labeled Metabolomics or Isotope-labeled Metabolomics. And by using DIMet, the questions about metabolite abundances and labeling speed can be resolved. And we have a level of pathway-based integration with other omics. We are also working to be able to automatize the network projection for combining both omics, metabolomics, and transcriptomics. Thank you very much for all these contributors or teams or collaborators in Belgium and the rest of Europe, and also for thanks to the Institute of Cancer and the GDR-BIM for the support for the registration to come here to this event.
Thank you for the presentation. You focused quite a bit on the technical details, how you implemented this tool, but I have to say I'm still struggling to understand how it actually works. Like, do you require to have a metabolic map for your organism? And can you use different labeled precursors than just glucose? Do you rely on some feature detection tools, or do you just focus on the raw spectra? How does it actually work?
Yes, I apologize. Maybe I went too fast on this scheme, and this figure explains what do we need. We need only or tables with the quantifications. No the raw data, no the peaks, no IMZML, no. But if you already have deconvolved your peaks and you have your quantifications, you only use your tables, and with a very modular approach, you can do all the analysis. It's not a black box. You can do the input and immediately get the output from specific modules without avoiding the confusion of what's happening in the middle. I don't know if that answered the question.
Okay, so you can use any kind of labeled precursor to, for example, discover new pathways where this label gets incorporated.
The work of the pathways is still the level of metabolograms is the user that defines the molecules that goes in the pathway. So the example shows this very well. And indeed, for doing the representation of a network, it's the work that will come in the future. It's not yet ready.
Thank you.
Yeah, maybe a little bit in the same vein. You would, for the multi-omics analysis, you would have on the one hand the metabolomics input, which can use different tools to generate it. Similarly, transcriptomics data, also different tools. Do you kind of have a documentation on GitHub or something about which input files DIMet then exist, so that people can transform the output from those different tools into input that DIMet understands?
Yes, precisely. In DIMet itself, we had the intention to offer this module, but as many platforms change of formats, we had to isolate this module in another different tool that it also has a GitHub, this trace groomer. For example, if your platform gives you the tables in Excel file, because Excel files are not accepted in DIMet, you can transform them via this tool.